It's a presentation by Emmanuel, Christine and Lilian on an article titled Insulin Gene Expression is Regulated by DNA Methylation. So DNA methylation is a biochemical process that is important for normal development in higher organisms. Insulin is a critical component of metabolic control. The article investigates the components of gene expression in both mice and in humans. So the methodology first. Pancreatic cells from two specific strains of mice were digested, incubated, and processed, leaving isolated islet cells. The pancreas contains clusters of cells that produce hormones, and these clusters are known as islets. The islet cells were cultured overnight and dissociated in order to individualize the pancreatic cells on their own so that they can be identified using immunocytochemistry techniques to determine their purity and composition. I will talk more about the immunochemistry later on. Human pancreatic beta cell sorting. 10,000 islet equivalent human islets were incubated with TREP-LE for 15 minutes. TREP-LE is used to dissociate the islet cell clusters from each other, so individualized cells are the results. These dissociated cells will then pass through 140 micron strainers. They're like this sieve you find in your kitchen. The cells were incubated with Newport green for an hour, which is used as a marker. They were then suspended in a cell sorter, and 10% of the sorted cells were used for immunocytochemistry, and genomic DNA was isolated from the remaining cells.
Told you to hold on to chat.
Okay, so immunocytochemistry. Immunocytochemistry is a technique that uses antibodies to target specific peptides or protein antigens. The following primary antibodies were used for immunocytochemical staining. Guinea pig, anti-insulin, and rabbit, anti-glucagon. The corresponding secondary antibodies that attached to each primary antibody were either conjugated with fluorescein isocyanate, which is the blue marker, or Texas red, which is the red marker, in order to see them under a microscope. And in this experiment, they used fluorescence microscopy to view the results and determine the cell percentages from the different colored markers. And this is an example of a fluorescent image. And a computer software was used to determine the cell percentages in this experiment. Okay, so cell culture. The mice cells were cultured in a cell incubator maintained at 37 degrees with atmosphere containing 95% air and 5% carbon dioxide, which are the typical conditions for mammalian cell culture. And variations in growth conditions can result in different phenotypes. The cells were passaged by tryptonization and subcultured on a weekly basis. In subculture, passaging is the process of transferring cells from one medium or cell culture to another in order to produce a large number of cells. As cells grow, adherent cultures tend to stick to each other or even to the inside of the vessel in which they are held. These cells need to be separated from each other and from the vessels in which they are held, such as in this experiment. It is commonly done with a mixture of trypsin EDTA, and a small number of the detached cells can then be used to seed a new culture while the rest is discarded. Next one. Bisulfide genomic sequencing. It's used to determine the pattern of methylation of DNA, treating it with bisulfide. The treatment converts some of the cystone residues to uracil while leaving some unaffected. And it's the reading of the changes between the cystole and the uracil which allows scientists to determine the differences in the methylation of the DNA. The results found there were no discernible differences in the tissue-specific myelinization patterns between the two types of mice tissues.
Change.
And these are some of the other steps that were used. Well, I'll go on to the next one.
6 bloody minutes.
6.
Jesus.
6 minutes, and we got 2 minutes to say our thing.
Okay, I'll cut that out.
Result and discussion. Okay. Fuck. Your part has put me off already. Previous studies have established the regulation activity at the transcriptional, post-transcriptional, transcriptional, secretary, and even post-receptor levels. In this study, they demonstrated that human insulin gene and mouse insulin 2 gene expression are also controlled epigenetically by methylation of specific CPG sites in the promoter regions. In non-insulin expressing cells, some of all of these sites are methylated, whereas in insulin expressing cell, they are completely demethylated. The sequence data revealed The sequencing data revealed that the 3 CPG sites in the insulin 2 promoter are uniquely unmethylated in pancreatic beta cells and in the insulin producing NIT1 beta cell line, but predominantly methylated in other tissues. As shown in diagram B, the insulin promoter in the insulin producing human beta cell fraction is unmethylated, but it is predominantly methylated in the beta cell depleted fraction. This result demonstrates that the adult human mouse insulin gene promoter exhibits a tissue-specific pattern of DNA methylation. The insulin molecule is central to metabolic homeostasis, and as such, insulin activity is regulated at multiple levels. In general, DNA methylation has a suppressive effect on gene transcription.
In conclusion, this study demonstrates that DNA methylation suppresses insulin gene expression, that the insulin gene promoter is specifically demethylated in insulin-producing cells. Methylation recruits MECP2 and possibly other methylated DNA-binding proteins, resulting in decreased binding of the transcriptional activators ATF2 and CREB. Furthermore, demethylation of the insulin gene appears to be an important and potentially rate-limiting step in the differentiation of insulin-producing cells from embryonic stem cells. Do you know what? Because